The overall goal of the following experiment is to determine intracellular or extracellular ascorbate levels in cultured or isolated cells. This is achieved by incubating cell extracts or whole cells with ascorbate oxidase to selectively deplete ascorbate in subsequent steps. In the first method, cell extracts are incubated with ferrocyanide to generate ferrocyanide in an ascorbate-dependent manner, which gives a direct representation of the amount of ascorbate in the extracts. Next, the amount of ferrocyanide formed in the cell extracts is determined by quantitatively oxidizing the ferrocyanide with added ferric iron to form ferrous iron, in order to generate a chromogenic iron 2 ferrine S complex that can be quantified spectrophotometrically. In the second method, whole cells are directly incubated with the iron 2 chelator ferrine S and ferric citrate. The results show differences in intracellular or extracellular ascorbate levels based on the quantitative and specific reduction of extracellular iron complexes that can be detected spectrophotometrically.
The main advantage of the current technique over methods that are already in place, like HPLC-based detection, is that this highly specific microplate assay can be carried out very quickly, and all samples and standards can be processed in parallel rather than in a serial manner.